You have to put your thinking caps on this morning, okay? It's a complex subject, and I'm going to do my best to boil it down to make it as simple as possible, yet at the same time, not make it too simple, because the complexity of it is really what's neat about it. So, to start off, I want you to imagine a world without life, okay? It can be hot, it can be cold, it can be muddy, but no life, nothing walking around, nothing breathing, living, nothing. Just kind of this soup. Can you imagine that? Can you kind of think of what that would be with no life? Not even microscopic life. I'm not even talking go into a microscope and see a cell and you see life there. I'm talking absolutely no life. But there are molecules, there are components, there are certain things that are there, there's matter there, but it's not living. So today, this morning, what we're going to tackle is how did it get from non-living to living? How did it go from abiotic to biotic? Now, scientists have tried to tackle this question, and they've come up with many theories. And today what we're going to do is look at these theories to explain this question, non-living to living. They're good after living. They can figure it all out after that. But how do we get life? And when I'm talking about living, I'm talking about a cell. So when you think living, think cell. And not to overcomplicate things, a cell is just a circle with stuff in it that kind of replicates and can do things, okay? And we'll get into a little bit more of what that looks like. So think cell, think living, okay? Non-cellular to cellular, how do we get there? So, to backtrack from cell, the meat of our talk will be DNA. And we've all seen DNA. We've seen this double helix, we see it all over the place. DNA, it's the blueprint, it's the kind of code. But to get a cell, you have to start with DNA. So I'll just draw a little squiggly line that's trying to represent the double helix that is DNA. And a DNA basically encodes a cell. If you're following along with your notes, the first thing, number 1 point says DNA encodes amino acids which form proteins. Okay, now don't get overwhelmed by this language. I'm going to help you make a parallel to what this means. So, if we have DNA forming amino acids which form proteins, and then proteins form a cell. Okay, so how can we help to think about this? If some of you are going, got it, I know it, it's a central dogma, we go from DNA to amino acids, proteins to a cell, I understand that. Some of you are like, what are you talking about? Well, this is to help you think about this, okay? DNA is like language, like the English language. Amino acids are like letters. So let's say these are like letters in our alphabet, okay? And let's say proteins are like words, all right? So letters make words, so amino acids make proteins. And then a cell would be like a paragraph, okay? A bunch of words coming together in a few sentences that make a paragraph. So let's call that a paragraph. So throughout the talk, when I'm talking about amino acids, proteins, cells, those sorts of things, you can kind of think of it like this. Think of amino acids as letters, think of proteins as words, and think of a cell as a paragraph, okay? Green, red cards, how are we doing? Can we kind of follow that a little bit? We're green? Okay, that's good. It's good. Okay. So again, the important information is, the important question is, where did this information come from? Where do we get DNA? If DNA ends up turning into living, ends up turning into cells, proteins, amino acids, where did this come from? Okay? And how have scientists tried to explain this? Science uses two principles. They use chance and necessity. And they use these two principles to explain life, to explain how we see things, to explain gravity, to explain all kinds of different things. okay? I think we understand what chance means. We're going to go a little bit more into chance. But we understand that chances are either good or bad. And we kind of understand that. Necessity, all necessity means is that if I drop a ball over and over again, gravity sorts of take effect, and the law of gravity necessitates that the ball falls. So basically, laws come up with things that happen to objects. So that's on the side. So we're looking at chance and necessity. This is how science kind of operates. This is how your future college professors will think about things. They'll think about the chance of something happening, and then they'll think about the necessity of it happening. So if I drop a ball, it's going to fall based on necessity. So, and that's your bullet point number 2. The 2 fundamental principles that scientists use are chance and necessity. All right, so what we want to look into first is chance. How well, in explaining non-living to living and getting DNA, does chance work? So soup, that world that was non-living into living, how well does chance work, right? And so what this guy, Albert Leninger, said, he wrote biochemistry, sold thousands of copies. He's kind of the grandfather of biochemistry. He says, we now come to the critical moment in evolution in which the first semblance of life appeared, living, through the chance association of a number of abiotically formed macromolecular components. All right, a lot of syllables there. Don't get caught up in the syllables. What Albert's proposing is he's saying that life arose from chance. Okay, how does this stack up? How does this stack up? Well, in order to demonstrate this a little bit, I want to use just a quick example. See how this works. Bananagrams, right? Everyone loves bananagrams. Oh, no. Open it wrong. Take a letter. There you go. Take a letter. Just grabbing letters here. 1, 2, 3, 4. Okay, that's good. That's good. I'll have to get my bananagrams later. Okay. Oh, so what word do we have? Why don't you start? What letter do you have? O? Okay. What do you have? L? A? E? R? Did I give anyone else one? Kyle, I'll give you one? I'll grab one. How about that? I got an I. All right. What do we got? Okay. I was getting a little nervous, to be honest. I mean, I had an O, a consonant, then a vowel, and then another vowel. It was actually starting to turn into a word, but not a word, right? I don't think so, at least, unless anyone's did. All right. How would I win this game, though? Let's say I did get caught, and I only used 4 letters, and it came to. How would I win the game? How would I keep going? I just kind of told you. I would keep going. I would keep pulling letters, and eventually it wouldn't be a word. Okay. So the question is, why didn't I get a word there? Chance, right? Kind of chance, but why doesn't chance work? Okay. There's a good way to explain it. It's a problem of combinatorials. Okay. Take a bike lock. We've all used a bike lock. A simple bike lock with 4 dials, 10 digits each. The possible combinations are 10,000. You have 10 times 10 times 10 times 10. It's 10,000 combinations. That's why bike locks work. You have one combination out of 10,000. So someone can't go up to your bike and steal your bike. There's 10,000 possible combinations with just a 4-combination bike lock with 10 options. Well, what about if you increase that to 10? So my picture doesn't work, but let's say it's now a 10-combination bike lock. Well, that's 10 times 10 times 10 all the way out to the 10th. That's 10 billion combinations, right? So we've gone from just changing the number of digits from 4 to 10. We've went from 10,000 possible combinations to 10 billion possible combinations just by changing 6 slots. We've added just 6 slots. So if you had a 10-combination bike lock, there's no way that someone could break into your bike. I mean, the chances of them breaking into your bike with 4 aren't very good either. That's 1 in 10,000. So, let's look at proteins. Here's the jump. If we can come back to our letters here, this didn't work because how many options did we have for the first slot? 26 English language, 26 letters. That's good. How about the second? 26, 26, 26, 26. And you don't add those, you multiply them. So the possible combinations, 1, 2, 3, 4, 5, 6, that's 26 to the 6th power. That's 26 times 26. That's a lot of combinations for just a 6-letter word, right? So the chances of us producing a real word were very, very slim based on those calculations. So that is the problem of combinatorials. But now let's think about amino acids, proteins, words, letters, those sorts of things. Now we need to make the jump from combinatorials, math, all that stuff, to the biology realm. So the question, how many amino acids, how many letters can be a part of a growing protein word? So how many, in our science language, not in the English language anymore, in our science language, how many are there? Well, there's 20. So instead of 26 letters, there's 20 letters in the science language. There's just 3, there's an example of 3, glycine, tyrosine, cysteine, leucine, valine, there's 20 of them. And if you want to major in college, you can learn how to draw all of those and memorize all 20 amino acids. It's not fun. So with that being said, 20 amino acids let's just take a small example. Let's take a small word. Let's take a small protein. 10 sites, so a 10 amino acid long word with 20 amino acids each. That's 20 to the 10th power. That's 10 trillion possible combinations for a 10 peptide, a 10 amino acid long word. Okay, how are we doing? We tracking? How about green and red? How are we doing here? We're greening? We all right? Okay, we're greening. All right. So, let's keep this in mind. This is a 10. This is a 10 letter, a 10 amino acid word we're talking about here. Okay? But here's the problem. A real protein is 150 amino acids to 400 amino acids long. All right, so this wouldn't function. In life, this is non-existent. A real protein is 150, so insert 150 there. And that number, I don't have enough space in this room to recreate this number, okay? So that's what a real protein sort of looks like. 150 sites, 20 amino acids long. That's 20 to the 150th power. That's equivalent to 1 times 10 to the 195th possible combinations. That's a lot of zeros. That's 195 zeros. Okay? Go ahead. Just to get one protein. That's just one protein. And remember, we're trying to get to cell, so we need a bunch of proteins to make up one cell. All right? So this is just one protein we're talking about here. Now, they've done calculations. They've figured that just as there was a possibility that we could have gotten a word, there also is a possibility that we could get a functional protein, right? I mean, some of those combinations work, fold together, look nice, and work. Well, Doug Axe, he's a big-time apologist, works at Caltech. He asked this question. He said, how common are those functional sequences? How common are those proteins among all the possible combinations? Well, he came to the conclusion that there are 1 in 10 to the 164. After he did the math, after looking at how many possibilities there were. So I did have some room here. This is 1 in 10 to the 164. That's your chances of getting a functional protein by chance. Okay? Again, here's the main kicker. Let me say this. We've been talking about one protein, right? One word, the chances. But it takes 250 to 400 proteins to produce a minimally complex cell. So it takes 250 to 400 of these things to produce one cell. All right, that's your bullet point number 4 if you're tracking. It takes 250 to 400 functional proteins to make a basic cell. And, oh yeah, we have millions of cells in our body. No big deal. Just to help you appreciate this, I'm pulling a clip from a documentary called Expelled. I'm going to be using a few clips from this documentary, and it's going to walk us into the inside of a cell, just one cell, and I hope you'll appreciate the complexity of it. Oh. 250. 400. So we traveled inside the cell. We haven't even got to the nucleus yet. We haven't even seen DNA. These are microtubules. They're working to move things along the cell. These are all proteins that are functioning to move things around in the cell. We have millions of these in our body, just cells. We haven't quite traveled to the nucleus yet, and here we go. We're going into the nucleus now where DNA is found. And here's DNA. It's all coiled up on these things called histones. And it's wrapped around, it's coiled up so it can store all the information. And these guys take the information from DNA, and they have to take it somewhere. They take it out of the nucleus to perform a function, to do something. And now, something's going to happen. So that's a cell. Pretty cool, right? All right. So based on this, scientists know this, okay? Scientists put these together. Scientists who don't believe in a creator and who have struggled to answer this question put this together. They all understand this. For this reason, and this is point number 3, I had to backtrack, no serious scientist thinks that life began on the basis of chance. They don't. No one believes this anymore because they realize the problem of combinatorials. They realize the chance is astronomically crazy, and they see this complexity. So if anyone, I need to go back. Okay. So basically we've concluded if you have a highly improbable event, the protein, which also performs a function, life, then chance is very, very unlikely. Chance is not a good hypothesis. So if anyone in school says to you, well, it all began by chance, don't get bullied there. Obviously, no scientist believes it, and I've just given you plenty of evidence for why no scientists believe that. So don't get bullied on this point. So from here, we need to move on and look at other options. So if scientists believe that chance was not possible, we need to look at other options for, okay, how do we get here? How do we go from here to here if, we use chance and necessity, and chance, we have to throw that out the window. I mean, they're not willing to say it was created, there was a designer. They need to explain, they need to use scientific rationale in order to get there, right? So our question was the origin of life, right? And Darwinism. You may be a little bit familiar with natural selection, Darwin, but just a real quick example of what natural selection and Darwin is, because scientists will use this in order to explain non-living to living. What natural selection says is that if you have let's go way back in time, a black chameleon, a chameleon that doesn't change color yet, it's only black, and it's still called a chameleon, all right? And it's producing, and it's going about its life, and then all of a sudden it has a baby that can change colors, that can suit to its environment. And now this baby that can suit to its environment and change colors has another baby, and they all can change colors. And over time, the chameleons that can change colors are living longer than the black chameleons, because the chameleons that can change colors, they obviously, that's a trait that they've inherited, that they've acquired, that makes it more beneficial for them to stay alive. So therefore, fast forward thousands or millions or whatever you want to say, how many years, we see chameleons that can change colors because the fittest have survived. Okay, the strongest have survived. That's Darwin evolution. You see the survival of the fittest in Darwin evolution and natural selection. Here's the problem. Well, before I tell you the problem, I'll tell you that natural selection depends upon the fact that things can self-replicate. Okay, so this chameleon that I was talking about, well, it could replicate, right? It could have a baby and then another baby, and it could continue with that process. So what scientists have said, what they've proposed, they know this about natural selection. They go, okay, we get it. Things acquire traits that make them stronger or make them better fit, and they continue, they go on. Well, what if we apply that same principle to this, non-living to living? It's more beneficial for molecules and this kind of soup. This is non-structured, just kind of a picture of molecules and whatnot. What if it went from this to living, to the DNA we saw, and is more beneficial? And you give it enough time, and it'll turn into something living. Does anyone see the problem with that? Okay, good. It's not alive, right? We went from non-living to living. In natural selection, Darwinism, it depends on self-replication. The whole theory depends on living. And so if natural selection presupposes organisms that can copy themselves, then we run into a philosophical problem that's called begging the question. Natural selection depends on self-replication, but self-replication depends on DNA and proteins. Okay? So what's the problem here? We went from non-living that can't self-replicate to living. It doesn't work. Okay.
So if it's non-living, why would it want to live? Because it's surviving.
That's the problem. That's the problem. Scientists have said if it's a soup, it's better not to be a soup. It's better to be DNA and cells that can self-replicate, that can form things.
But why, if you have natural selection? Like, why would you want to be living and possibly die when you're not living? You're not dead.
So why would molecules want to be living?
Yeah, if they're perfectly fine surviving.
Right. So but they're not living. Wouldn't you rather live than not? Wouldn't you rather be able to reproduce yourself and be able to be more complex? And that's better. It's better to be living than dead, right? And from a scientific perspective, you're active, you're engaging with the world, okay? So because of this, because of the problems that this kind of churns up, and especially the non-self-replication part, scientists have scrapped this idea too. They've said natural selection, Darwinism, man, we would love to be able to apply this everywhere across the spectrum, but we can't because we can't go from non-living to living. It can't self-replicate. So, when you look at kind of the last example of how scientists have tried to approach this problem, and this is the example that's gotten a little bit more steam. People are talking about this a little bit more. And this is a guy by Dean Kenyon. He wrote Biochemical Predestination. That has some theological weight to it, if you ask me. So what Dean has said was that molecules in our universe sort of have an attraction to each other, just naturally. If you throw compounds just together, they like, certain molecules like each other a little better than other molecules. Can anyone think of two components that if you throw them together, they form something? It's tough. I couldn't get this. What's that? That's good. H2O? We're thinking that? That's good. That's really good. That's even better. The example I had was salt, sodium and chloride. You throw salt together. You said it back there? Oh, did you say it or not? Oh, all right. You're just excited. Okay, that's good. So salt, boom, voila. If you throw sodium, chloride, a bunch of other chemicals, we can make salt. And, so it's not a crazy idea. Dean saw this, and he said, well, what if the same thing applies to DNA? He asked this question. He said, is this salt, it's beautiful. It's crystal. This forms on its own. It takes no energy. It takes nothing to form that. Those are ionic bonds right there. So is this like this? This is DNA. Dean has said, well, possibly if you throw enough components together, they'll kind of self-aggregate, and voila, we have DNA. But we run into a problem. The more we found out about DNA, the more we analyze its structure, we realized there was a problem. So, here's a magnet board. I have a little message. Hi, Apex. Okay. What happens if I do that? Okay. So, question to you, is there any attraction between these guys? right now on this magnet board? Is there an attraction to anything else? To the board, right. So there's an attraction there, right? This is a picture of DNA. If you look at the structure of DNA, we have a backbone. We have a phosphate backbone, and it runs along each side of DNA. That functions as a magnet board, all right? And indeed, there are attractions. The information, the letters in DNA are attracted to the backbone. They love it. It's how they make their living. However, there's no attraction within the backbone. There's no attraction between the letters. There's an attraction to the backbone of DNA, but there's no attraction between the letters themselves, much like this. So Mr. Kenyon has since kind of gone back on his theory. It got a lot of weight, and it got a lot of movement in the 80s, and people were kind of thinking, hey, that makes a lot of sense. But since we've understood more about DNA, we realize, man, there's no attraction between the letters either. So you remember the message I had up there. How did that message get there? So I did it. So it didn't just spontaneously, I didn't pick this up and it said hi. No. I put something into it. Just something to think about. Okay. We're fast approaching the end. and I want to say this, that there is no naturalistic explanation for the origin of the information that was used to build the first life. And by naturalistic, I mean what science uses, what your college professors will use. They'll use chance, they'll use necessity, they'll use Darwinian evolution, they'll use self-aggregation with molecules. There is no naturalistic explanation for the origin of life. And for that reason, scientists have kind of gone a little crazy. And I'm going to show two clips here. These are the leading scientists of the day that they know this, and they can't. I think the clips speak for themselves.
The chemical elements essential for life, hydrogen, oxygen, carbon, and nitrogen, were now in place. What was needed was a way of combining them. Perhaps the energy came from lightning. Whatever it was.
Excuse me?
Whatever it was, energy managed to arrange these chemical ingredients in just the right way.
Whatever it was? I was hoping for something a little more scientific. The most popular idea has been that life emerged spontaneously from primordial soup. In 1953, Stanley Miller mixed water, methane, ammonia, and hydrogen to simulate the early Earth's atmosphere. Then he ran electricity through it in an attempt to jumpstart life. It didn't work. While the initial results seemed promising, 50 years later, most serious scientists have abandoned this approach in favor of alternate theories. Prominent Darwinist Michael Roos attempted to explain one of them to me. He wasn't kidding.
How did we get from an inorganic world to the world of the cell?
Well, one popular theory is that it might have started off on the backs of crystals. Molecules piggybacked on the back of crystals forming, and that this led to more and more complex. But of course, the nice thing about crystals is every now and then you get mistakes, mutations, and that this opens the way for natural selection.
But at one point there was not a living thing.
Yeah.
And then there was a living thing. How did that happen?
Well, this is I just told you. And I don't see any reason why you shouldn't go from very simple to more and more complex to more and more complex.
I don't either, but I don't know how you get from mud to a living cell. That's my question.
Yes, well, I've told you.
I think it was on the
try
back
one
of
more
crystals.
time.
On the backs of crystals.
On the backs of crystals is at least one hypothesis, yes.
So that's your theory. You think that is more likely and less far-fetched than intelligent design?
I think it is.
Okay. That man was Michael Roos. He's a philosopher of biology at Florida State University. He's well known for his work on creationism and evolution controversy and has written many books on Darwinian evolution. If that wasn't enough, we're we're going to look at one final slide. And you want to talk about sort of the kingpin of all science. It's this guy, Richard Dawkins, especially from an atheistic, non-God kind of view. He's a British guy. He's written a lot of books. He's written The God Delusion. And we're going to look at a final clip that Ben Stein used to ask Richard Dawkins this exact question. How do we get here? How do we get here, Richard?
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
What was that?
It was the origin of the first self-replicating molecule.
Right, how did that happen?
I've told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else. What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility.
Talking aliens here.
Intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of design.
Signature? Okay.
Wait a second. Richard Dawkins thought intelligent design might be a legitimate pursuit?
And that designer could well be a higher intelligence from elsewhere in the universe.
Well,
but that higher intelligence would itself have had to have come about by some explicable or ultimately explicable process. It couldn't have just jumped into existence spontaneously. That's the point.
So Professor Dawkins was not against intelligent design, just certain types of designers, such as God.
So the Hebrew God, the God of the Old Testament, he doesn't exist in your view?
Certainly. I mean, that would be a very unpleasant prospect.
And the Holy Trinity of the New Testament?
Nothing like that.
Do you believe in any of the Hindu gods? Like Vishnu?
How can you ask such a question?
You don't,
how
right?
could I? I
Do you
mean,
believe
why
in-
would I, given that I don't believe in any others?
You don't believe in the Muslim God?
No. And why do you even need to ask?
Well, I just wanted to be sure. So you don't believe in any God anywhere?
Any God anywhere would be completely incompatible with anything that I've said.
I just wanted to make sure you don't believe in any God anywhere.
No.
What if after you died you ran into God and he said, "What have you been doing, Richard? I mean, what have you been doing? I've been trying to be nice to you.
Yeah.
I gave you a multi-million-dollar paycheck over and over again with your book, and look what you did."
Listen to this.
Bertrand Russell had that point put to him, and he said something like, "Sir, why did you take such pains to hide yourself?"
To hide yourself. Richard Dawkins has seen the video that we looked at going into a cell. Say, God has hid himself from him. Folks, that's all I have. Maybe we're looking at something that appears designed because it really is. Scientists are unwilling to come to this conclusion, but any rational mind, I think, would. So. Yeah, right.
Are those present anywhere else except for in DNA? Like, could someone look at something else and see those?
Right. So the guanine, taurine,
Like,
those things.
Are those present outside of DNA anywhere else? Or can you put them together?
The-
Or are they solely in DNA constructed on it?
They're solely in DNA. They're only found in DNA. The cell is constantly trying to salvage those things. So when a cell dies or whatever, it'll take those certain bases and throw them into DNA. Again, I mean, it's amazing. So yes, yeah.
I just wondered if it was like only DNA.
Yes, it's only found there, right.